In this video, we'll be looking at hemostasis. Now, hemostasis basically is a fancy way of saying stopping or limiting blood flow in terms of bleeding. And of course, if you really want to think about the concept of hemostasis, it's one of those things where you need it to happen fairly fast. It has to happen in an appropriate situation. You don't want, say, things like blood clots to form when you don't want them. And once you do get to something like a blood clot, you have to have mechanisms to basically disassemble it after it's no longer needed. Now, before we get to blood clots, we're going to have to talk about platelets, and we're going to have to talk about some of the reactions in the body that get you to forming a blood clot. So hemostasis is, there's the word right there, hemostasis, is basically stopping bleeding because of potential of fatal leaks, hemorrhaging. Now there's three mechanisms, and we're going to look at all three. We're going to start with a vascular spasm. We're going to also talk about platelet plug formation, and also blood clotting. Now blood clotting will be in the second part of the video. Now let's start by talking about platelets. Now platelets are interesting because they're not technically a cell. They're actually a small fragment of a cell called a megakaryocyte. And we're going to talk a bit about the functions of them. We're not going to get hung up on the numbers here. But the functions are that they do a number of things. They secrete vasoconstrictors that help to reduce the blood loss. They also have the ability to stick together once they're activated and form platelet plugs, and that helps to seal very small breaks. They also secrete procoagulants, and this helps to promote the clotting. Also, not only do they have a role in initiating clotting and stemming blood flow, they also have a role in resolving blood clots. So at the appropriate time, they can be involved with clot-dissolving enzymes formation. In addition, they can call in some of the white blood cells, such as neutrophils and monocytes to the site of inflammation. And the list just keeps going on. They help to phagocytize and destroy bacteria, and also they secrete growth factors to hopefully repair through mitosis the blood vessels. So you can see that altogether the functions of platelets is quite extensive. Let's talk a bit about them. Now, they're not a cell, which is why often we refer to the blood cells not as cells. You know, red blood cells are cells, white blood cells are cells, but platelets are not. They're actually a fragment. And they come from this large cell right here, which is called a megakaryocyte. Now you can get a sense as to how big. This is a megakaryocyte right here. Here's a red blood cell. Here's a white blood cell. You can see that it does deserve its name, mega for huge. And what you can see here, the basic mechanism is that there's like finger-like projections of the megakaryocytes, and it's off these finger-like projections that the platelets arise from. They basically form, break free, and off they go as platelets. Now, when we're talking about platelet plug formation, or I should say platelet formation, it's thrombopoiesis is a fancy name for platelet production. And there are stem cells. Excuse me, that just hopped for a second. There are stem cells. And we're going to start just by quickly talking about the megakaryocytes. And what they do is, look at the name, mega, they're huge, karyo for basically nucleus DNA. What they do is they repeatedly replicate their DNA, but they don't divide. And they end up forming this gigantic cell known as the megakaryocyte. And it tends to, you can really see it has a bilobed nucleus. Now, megakaryocytes, they live in the bone marrow, and they're adjacent to the blood sinusoids. And this allows them, because remember there's fairly large gaps there, it allows them to basically have their almost finger-like projections dabble into the blood that we saw before. And they have these long tendrils of cytoplasm, and they're basically the proplatelets that they protrude into the blood sinuses. And the blood flow splits off the fragments, which we call platelets. Now, how long do they go for? They exist for about 10 days. And there's actually some places where they're stored. 40% are stored in the spleen. Now let's quickly talk about three main mechanisms. We're going to look at vascular spasm, platelet plug formation, and coagulation. But let's just start with the first one, which is the vascular spasm. What happens here, the name tells you everything you need to know, basically. What happens is that the vascular spasm causes a constriction of the broken blood vessels. This helps to limit blood flow, and we'll talk a bit about that. Now, what causes it? That the pain receptors seem to have a role in it. Also, the injury of smooth muscle. Remember, smooth muscle can react to a lot of stuff in order to constrict. And they also release serotonin, which is a vasoconstrictor. Now, what are the effects? The effects are that I get a fairly quick constriction of the broken blood vessel. Now, the pain receptors tend to keep that going for a few minutes, and then the smooth muscle injury is sort of a more longer duration effect. Now, what's this all doing? All of this is basically buying us time and setting up the conditions for the next things to happen. The second thing that happens typically after this is I end up with platelet plug formation. Now remember, the lining of the blood vessel is coated with almost a Teflon. It's coated with prostacyclin. It's a platelet repellent. But as soon as you disrupt the inside lining, then what happens is you've exposed collagen. And the platelets can have little pseudopodia, little false feet, that can stick to the damaged walls, and they'll start to pile on each other. And the platelets can actually reach out. So, you have a platelet here. It can start to reach out a pseudopodia, and maybe it can reach out in a couple different directions, which allows it to grab on to, say, part of the wall. And then it can start to pull it in, or it can connect with other platelets and interact with them also. So the platelet pseudopodia, they contract and they draw the walls of the vessels together, forming a platelet plug. And then they also degranulate, and they release a number of substances. One is, once again, as I said earlier, serotonin, a vasoconstrictor. ADP attracts and degranulates more platelets, so you almost get a bit of a chain reaction happen. Thromboxane A2, it's an eicosanoid. It also encourages platelets to aggregate there and to degranulate, and more vasoconstriction. And if you think about it, this is not a negative but a positive feedback cycle, and which is what we want, because we want things to happen in a big way very quickly. Now, the next thing will be coagulation, and we're going to talk about coagulation in the next video.